Okay, so for this audio lecture, we're going to be looking at microbial pathogenicity. So the three kind of subsets to this would be, we're going to be looking at relationships. This is covered in Chapter 4, Section 1 of the OpenStax Microbiology textbook. Also pathogenicity, which is in Chapter 15 of the OpenStax Microbiology textbook. And then also virulence factors, and that's going to be Chapter 15, Sections 3 and 4. So Section 3 focuses on bacteria and viral, and Section 4 is miscellaneous microbes and some of the virulence factors for that. So when we look at microbial pathogenicity, we have to keep in mind a lot of times when we're talking about the host, we're talking about mammals, so humans, but other organisms can be host for microbes. And when we look at these multicellular organisms, they have pretty elaborate physical and chemical and also immune defenses to protect them against microbes that can cause disease. And if you've taken like an intro micro class or a anatomy and physiology class, you would have learned about the immune system, also about the normal physical structures of organs like the skin, also chemical defenses that we have to protect ourselves against these disease-causing microbes. But there's weaknesses, and so good pathogens, microbes that can cause disease, will exploit these weaknesses. And when they exploit these weaknesses, this can lead to the tissue having abnormal physiology. So thinking about anatomy and physiology, when you learn about different organ systems, you learn this is how it normally functions, this is what it normally does, this is how it maintains homeostasis. Well, if you have infection in there, if you have an overgrowth of microbes, it's not going to be able to function like it normally would. And because of this dysfunction of the tissue of the organ, this is going to result in signs and symptoms and result in disease. This disease can have short-term effects. It could have long-term effects depending upon the damage that's done to the tissue and how it can function. Okay. And so again, microbes have different factors, virulence factors, that allow them to take advantage of our defenses, and in some cases even use our defenses against us. And so the question really becomes with microbial pathogenicity is, how do these very small organisms that we sometimes need microscopes in order to see, how can they be pathogenic against us? We're multicellular. How can they take us down? And so we're going to look at some of those pathogenicity factors. And our understanding of these different factors is increasing. So as you guys are seeing in our journal club papers that we're covering over the next couple weeks, looking at bacterial pathogenicity, viral pathogenicity, fungal pathogenicity, we can see some of these different factors and how our understanding has changed about these small little organisms and how they can work together. So things like quorum sensing is starting to look like that's really important, and how some of these factors are turned on, how they share the genes that are responsible for these different factors also is really interesting when we look at sharing of plasmids, translocations of genes that can happen. So when we're looking at pathogens, again, we need to have a host for them. And so really we're talking about symbiosis. And so symbiosis is when we have two organisms living together. We tend to think of symbiosis as being this pleasant experience where host and other organism is living in a positive manner, but really this is more specifically mutualistic or commensal when it's positive. Symbiotic relationships also include negative relationships. So again, sometimes that term symbiotic is has this connotation of being positive, but we don't want to forget that there are negative relationships. And so this table from the textbook looks at the relationships that can arise. And so again, we can have things like mutualistic, where both organisms are going to benefit. And so our gut microbe, really, it's a mutualistic symbiotic relationship between us and our gut microbes. So they benefit because they have somewhere to live, we feed them, they have food, and they have relatively little competition. So they have competition among the other gut microbes, but compared to what they could have to deal with in other environments, not so bad. We benefit because they produce vitamins for us. They help to digest our food, so there's more nutritional value for it. They also are responsible for some of how our immune system learns, gets educated, and develops. And so truly it is a mutualistic relationship. Some textbooks, even like 5 years old textbooks, they refer to as gut microbes as commensal because we believed that the microbes were benefiting. There was nothing that they were doing, for us. We weren't benefiting from it, but really that's not true, and that's not what evidence is showing. Evidence is showing really gut microbes fall into the mutualistic category. To the other extreme, we have parasitism. So this is where we have a parasite that's going to benefit from the relationship, and it's going to harm the host. So this is where we'd have a pathogen. Pathogen is going to benefit because it has a place to live, and it actually damages the host. And again, this is where we start to see disease state where normal pathology of the tissue, normal function of the tissue isn't happening. So some of the terminology that I've been using with pathogenicity. So again, host-pathogen interactions. So we are going to have hosts. We tend to think of humans as being the host, but we want to keep in mind, if we're talking about veterinary medicine, the host could be another organism. So it could be a dog, a cat, a horse, or if we're doing exotics, it could be a tiger. If we're talking about botany, the host could be a plant. So there's going to be pathogens that are pathogenic towards plants. So plants are susceptible to pathogens that are bacterial, that are viral, protozoans. They can have ectoparasites, so they can have parasites, pathogens that live on their surface. For going back into a mammal host, we would think of things like a leech or a flea or a mite as being ectoparasites. We can have endoparasites, these live inside the host, and so bacteria would be an example. Viruses, but keep in mind that viruses have their own term. So viruses are known as an obligate intercellular parasite. So obligate require intercellular inside the host cell. So viruses aren't able to reproduce. They can't make copies of themselves without a host cell. So they're using the host cell's machinery to make copies of all their proteins, of their nucleic acid that they need, and then that gets released by the cell. And that comes at a cost to the host cell, so truly parasitic. Again, but viruses are non-living. And again, keep in mind with the term infection, this is where we're talking about some kind of damage to the host, the normal function of the tissue. Individuals going to start showing signs and symptoms. The reality is we're all walking around with subclinical infections where there's microbes present, or microbes are present at higher numbers than normal, but yet they're not causing damage yet. So a true clinical infection is going to have signs and symptoms of that. And at that point, the pathogen has increased in numbers. When we look at the virulence of a pathogen, there's a couple different indicators of how virulent a pathogen is. So there can be controlled experiments that are done in laboratory animals where we can find out what the median infectious dose is and median lethal dosage. So with median infectious dose, we're looking at how many cells, how many virons, how much of the pathogen is required to cause an active clinical infection in 50% of the population. So if this is a controlled experiment in laboratory animals, 50% of the inoculated animals in the study. So again, this is where the host has an infection showing signs and symptoms of infection. For lethal dose, this is what's the number for 50% of the infected animals, infected hosts, is to die. So really extreme negative outcome. And so keep in mind that infectious dose is going to be different than lethal dose. So lethal dose tends to be higher. Now this table shows you the infectious dose for some of the foodborne illnesses. So it's broken down into the different categories of pathogens. So you can see first we have our viruses. So you see like norovirus, as small as one particle, will give us 50% of those individuals becoming infected with norovirus. So they would have signs and symptoms of it. They could transmit it to other individuals. For bacteria, they tend to need a higher infectious dose. So if we look at something like E. coli, we can see that for hemorrhagic, which is infectious, pretty infectious, we would only need 10 to 100 bacteria. So imagine a hamburger and there's 10 E. coli on it, and if it's the hemorrhagic strain, we could then become sick. 50% of the individuals consuming that hamburger would become sick. If it's a different strain of E. coli, the dosage is much higher. So you can see some of these strains, you would need over 10 million bacteria in that material consumed. So much higher dosage required in order to get illness. And so you can see that even within a genus and species of bacteria, different strains are going to have different infectious doses, and they would have different lethal dosage because of the virulence factors, the genes that they carry. When we look at Salmonella, you can see we need over 1,000. Shigella is relatively infectious, so between 10 to 200. We also have protozoans, so you can see Giardia. Just a single Giardia can lead to an infection in 50% of individuals. So when we look at pathogens, they can fall into two different categories. We can have primary pathogens. Primary pathogens will cause disease in the host regardless of their health. So even healthy individuals can get disease from a primary pathogen. So their resident microbiota, their immune system is not going to be sufficient. The virulence factors that the pathogen has are high enough that, again, if you have kind of normal exposure to it, you will become sick with it. So Shigella is an example of a bacteria that causes dysentery, diarrhea. And so if you consume contaminated food or water that has Shigella in it, you're pretty much guaranteed you're going to get sick. It's a primary pathogen. With an opportunistic pathogen, this is where the host factors into this more so. With an opportunistic pathogen, it tends to be individuals with compromised immune systems. So their defenses are not fully functioning like a healthy individual, and so they're going to be more likely to succumb or to get illness from these opportunistic pathogens. Things that can compromise a host defense: if someone's been put on immunosuppressive drugs, if they're undergoing chemotherapy for cancer treatment, if you have someone who, because of a viral infection like HIV or AIDS, they have a compromised immune system, so they don't have a functional adaptive immunity. Also, there's different genetic mutations that can lead to individuals being compromised, and even things like pregnancy can put individuals at a compromised state, so they're more likely to become sick. So something like listeria, for a healthy individual, tends not to be an issue, but for an individual who's pregnant or a young child, listeria can actually become serious. So not just in illness, but also in death. And so that's one reason why pregnant women are told, not to consume. You hear about don't eat, deli meats, and watch what cheeses you eat. Make sure it's pasteurized cheese that's made with pasteurized milk, because there's a potential for listeria present. Now, when we look at virulence factors, we want to keep in mind that there are unique virulence factors for each individual pathogen. And so depending upon what virulent factors they have, either individual virulence factors or combinations of virulence factors, that's going to determine the extent and severity of the disease they cause. And so again, we just saw that E. coli, there are certain strains of E. coli that the virulence factors they have causes them to be extremely infectious and even lethal. Where other strains, because of the virulence factor they have, they don't have the same extent and severity of disease. Now, virulence factors get coded by genes, so we can look at the genes to identify which genes are responsible for the virulence factors, and that's where a lot of the research has been done in the last, I'd say, probably decade, where they've looked at what genes correlate to these different virulence factors, some of which we've talked about. Also looking at things like antibiotic resistance and such. There's genes that get transmitted. They can get transmitted through plasmids. They can get transmitted through these pathogenicity islands that can translocate to different areas on the chromosome. But again, we can look at these genes, we can do mutations of these genes, we can delete the genes to really demonstrate that this gene is responsible for the virulence of this pathogen. So we can also look at inactivating these factors. So maybe this is giving an inhibitor, so a drug to the individual to deactivate, to neutralize these virulence factors. So in our journal club paper with the fungal virulence factors, with that enzyme, we saw how they had inhibitors that they could give the mice, and that actually caused the immune system to be able to respond because the immune system wasn't being deactivated in that case. So again, some of our future drug treatments and even current drug treatments target these virulence factors of these pathogens. So first we'll look at bacterial virulence factors, and they fall into three main categories. So we can have virulence factors related to adhesion. So bacteria need to bind to host tissue cells in order to then be able to colonize the individual. And so there are molecules that allow for adhesion to happen. We can also have bacteria producing toxins, and they fall into two main categories, exotoxins and endotoxins, so we'll talk about those. And then we also have invasion of the immune system, and so if we think about the immune system, we have innate and we have adaptive, and so we can have different factors that target different parts of the immune response, so we'll look at those too. So this table is from the textbook, and it looks at some of those adhesion molecules that allow for the pathogen to bind to the host. And so these adhesion molecules have specific targets, and those targets relate to the tissue, the cells, the host cells that they want to target, that this pathogen ideally can live and survive in because of the environment in there. So again, these adhesion molecules will bind to receptors, molecules on the surface of these different cells to allow for them to be able to attach. So again, they can end up staying there, they can end up increasing in numbers and cause infection, and then they can produce something like toxins to cause damage. Okay, so we see that we have some streptococcus here that produce adhesion molecules. We also have E. coli and Vibrio, and again, these all are going to produce adhesion. You'll see fembrii for the E. coli. It produces fembrii, and that fembrii acts as attachment to those intestinal epithelial cells. So next, looking at the toxins being produced, we want to keep in mind that not all bacteria produce toxin. So some of the terminology that we want to keep in mind is with bacteremia, this is where we have bacteria in the blood. That's different than if we have viruses in the blood, so viremia. It's different than if we have toxins in the blood, toxemia. So again, each one of these is different because it's what part is in the blood. So a bacteria could produce a toxin, and the toxin gets in the blood, but the bacteria is not actually in the blood. Now the case is the bacteria moves in the blood, uses the blood, the circulatory system, as a highway to get to other locations, to secondary tissues to cause infection. Now it's possible that the bacteria is present in the blood and it's actually multiplying, so this would be septoremia, and so this can actually lead to septic and shock. So if you've heard of someone going septic, this means again that the bacteria is in their blood, it's multiplying, and again, because it can use the circulatory system as a highway to all these different tissues, it means it can get into different tissues and cause injury there. And so septic shock can be life-threatening. It can cause a decrease in blood pressure, and then this can lead to cells and organs being deprived of oxygen and nutrients, and so then the tissues can actually end up dying in the process. When you go into septic shock, it's not usually one organ system that's affected. Multiple organ systems can be affected, and medically we're not very good at treating multi-system organ failure. We're better at treating a single organ system, and that becomes important when we talk about endotoxins and exotoxins in a moment. Okay. When you go into shock, when you have bacteria in the blood and multiplying, sometimes they can release toxins present, and those toxins can be a virulence factor because they can cause damage, and that can further lead to low blood pressure, and it just compounds upon each of them, and that becomes really difficult to treat. So looking at some of the exotoxins, another table from the textbooks looks at some of the different exotoxins that are produced in some different pathogens. So you can see Staphylococcus aureus has a couple different exotoxins that it can produce. These are enzymes that can degrade either the matrix or tissues holding the cells together, so that can also degrade the DNA, and so it causes damage to that host cell, so the host cell can't survive. The Clostridium also produces proteases, so this is going to break down some of the connective tissue and proteins. So you can see in the figures on the right how these different enzymes, these exoenzymes that get secreted out of the cell, out of the bacteria cell, can damage the host tissues, and again it would affect the normal physiology of the tissue. And again, if these then can also make it into the bloodstream, that can become an issue. Now, one of the good things is that these exoenzymes tend to be specific, so they have specific targets. They don't affect all cells. So the other toxin we're going to look at is endotoxins. So for endotoxins, this is actually a breakdown of lipid polysaccharide. So gram-negative bacteria have that outer membrane. So we are going to have a cell membrane, a cell wall, and an outer membrane. That outer membrane has lipid polysaccharide. And so that structure that you see in the top of this slide is lipid polysaccharide. And so you'll notice that there's a lipid A, there's a core, and an O antigen. Now the O antigen is seen by the immune system. And if the immune system sees this, it's going to target the O antigen and actually causes the lipid A to be released. The lipid A can also be released when the gram-negative cell dies, either naturally or because the immune system is able to target it. And this lipid A can get incorporated into cells that have lipids. And so if you think back to like an intro bio class, all cells have lipids. And so lipid A can incorporate itself into many different cells inside the host. So luckily at low concentrations, it doesn't tend to have as much of an effect. The immune response is actually able to kind of regulate that through inflammation. But at high concentrations, because it causes so much inflammation, so much damage to tissues, this can relate to septic shock. This can cause a severe drop in blood pressure, multi-organ failure, and again, death. So with gram-negative bacteria, there's a concern about this endotoxin, this lipid A being released, and again, especially at high levels of it. So when we compare and contrast endotoxins and exotoxins, endotoxins, again, keep in mind this is coming from the outer membrane. So you'll see some sources call it the cell wall. It's not the cell wall. It's the outer membrane from that LPS, so it's that lipid A portion, and it's gram-negative bacteria. So gram-positive bacteria do not produce endotoxins. So if you have a gram-positive infection, you don't have to worry about endotoxins. As I mentioned, endotoxins can be systemic because they can affect any cell that has lipids. This leads to inflammation. A sign would be the individual having a fever. And lipids tend to be heat stable, so even with the fever, they can still be stable. Now, lethal dose for exposure to endotoxins is relatively high, so again, you need a high concentration to end up having an effect. But as we're seeing a growing number of gram-negative bacteria become resistant to antibiotics. Some individuals are going to have infections with high numbers of gram-negative bacteria. So once they start dying, they're going to release the lipid A, which then becomes an issue. With exotoxins, they can be produced by gram-positive or gram-negative bacteria, mostly gram-positive bacteria. So it's easy. It's kind of easy to think about, like, okay, endotoxins are gram-negative, exotoxins are gram-positive. But just keep in mind that there are gram-negative bacteria, like E. coli, like Shigella, that do produce exotoxins. Exotoxins are proteins. So this is a string of amino acids. So we need protein synthesis to occur. The cells need to be alive in order to make proteins, in order to make exotoxins. So the great thing is, if you can kill the bacteria, they can't produce exotoxins. Other thing is, because it's proteins, it's possible that we can have a neutralizing antibody against it. So if you know someone has an infection and that there's concern that the exotoxin is going to be an issue, if we have a neutralizing antibody, we could use that as a treatment. Okay. When we look at exotoxins, because they're proteins, they tend to have specific damage. So this target cell needs to have a receptor to bind that exotoxin. And so usually an exotoxin only works against a single cell type. So it's not going to be systemic, it's going to be specific. So we usually only have a single cell type, a single tissue, a single organ being affected. It's not multi-system. And proteins denature. And so if an individual has a fever, that can deactivate exotoxins. So they're not stable with heat. There are some exceptions to that, but again, generally, exotoxins as proteins are not stable. Now, unfortunately, lethal dose is going to be low because it's specific. So again, if it's not caught in time, enough damage is done, and there is not an option for something like dialysis or organ transplant, then the issue of an organ failing can be lethal. So with some toxins, these are exotoxins that are produced, they can act as neurotoxins. And so we can see how with these neurotoxins, they actually are going to block normal receptors. And so the normal, like acetylcholine, that normally would act as a neurotransmitter and cause muscle contractions, botulism toxin and tetanus toxin can actually block these receptors, so we don't get the normal stimuli that would happen. So this actually causes paralysis to happen. So again, some of these toxins, because they're acting on specific receptors, have a very specific response. All right, and so for the next couple slides, we are going to be looking at virulence factors that look at immune evasion. So if the pathogen can evade the host immune response, it's going to be able to survive. This really becomes important because if the pathogen is going to increase in number and cause damage, the immune system is going to be looking for them. There's going to be signs that factors the tissue is going to produce, like chemokines, signals to the immune response that say, hey, I'm being damaged, please help me. So cells can produce cytokines like interferons to signal that they're virally infected. They can produce chemokines, factors that signal that there's bacterial damage done too. And then there's also, so there's neutralizing those factors, but then there's also production of different components and different structures and different enzymes that also help to invade immune response. So our first example is looking at capsule. So the capsule is a coating around the bacteria, and so this actually makes it tough for phagocytes, immune cells, to actually attach and engulf pathogens, bacteria. And so when we see bacteria producing capsules, it actually correlates to them to being able to evade the immune response. Some pathogens will produce proteases that actually will kill the they're either capable of killing immune cells, or they can actually break down antibodies, so the antibodies aren't going to be effective. We can also have bacteria that are going to have fimbriae present, and so this is going to allow for them to attach, but it can also allow them to block phagocytosis from occurring also. Another example where we can block phagocytosis are acid-fast bacteria. So mycobacterium have a macolic acid, this waxy substance, on its cell envelope, and that makes it difficult for phagocytes to engulf those. And even if they are engulfed, this acid coat actually can cause the phagosome not to be able to digest them. So if you think back to phagocytosis, you're going to get these lysosomes with digestive enzymes fusing with the pathogen. And so, again, this waxy coating actually stops it from being digested, broken down. There are Staph aureus, that are able to produce an exoenzyme called coagulase. So coagulase actually causes clotting to form. And so what happens is the bacteria gets trapped in the clot, and it can hide out there. And so it's able to actually survive in there. Then what they can do is produce kinases, and the kinases will actually dissolve the clot, and so it allows them to then escape. So the immune system is like, oh, we trapped it in the clot. Well, we can't get to it, but at least it's in there. But then they can get out with the kinases. And so there's other, it's not just Staphylococcus aureus that has this strategy. There's also different streps that are able to digest clots. So again, a normal immune response would produce clots, but Staphylococcus aureus with coagulase can produce excessive clots, which can become an issue because those can cause clots leading to stroke, heart attacks, and whatnot. And then we can also have kinases being produced to dissolve those clots so the immune system isn't able to trap them. I think of the clots as like little spider webs trapping the pathogens normally. Then we can also have where we have anagenic variation. So the pathogen can actually alter surface proteins. Sometimes these surface proteins look very similar. They have high homology to our human proteins or human glycocalyx glycoproteins on our surface of our cells. So it becomes difficult for the immune system to distinguish them. Also with these changes in variation, it can be meaning that an antibody that used to work against the pathogen won't be able to recognize this new variant, this new anagenic variation. So this figure just shows you some of the different mechanisms. So you can see how we have this non-capsulated bacteria in panel A. And so that would be easy for the immune system to be able to attach and engulf through phagocytosis. But when we have the capsule around those, it protects it so that we can't get that direct recognition of PAMPs, of these molecular patterns that are found in pathogens. And so because we can't get those interactions, we can't get phagocytosis. In the middle panel in B, this is where we can see the capsules actually interfering from the antibody being able to recognize the antigen. And then on the last panel, panel C, we can see the proteases that are produced actually break down the antibody. So even though the antibody initially recognizes, it's not long-lived because the protease is able to break down and cleave the antibody, so it's not going to be active. With viral virulence, we're going to look at viral adhesion, so different molecules that are found in viruses that allow them to adhere attached to host cells that give the host range for a virus or a pathogen. And then they also, viruses tend to exhibit viral variants or viral anagenic variations. So similar to bacteria where they can change their surface proteins, viruses are able to change their surface proteins so they look new and different. So again, antibodies wouldn't work. But I just do want to acknowledge viruses do have other mechanisms at play. Some of those are, one of those we'll be looking at in our journal club paper with measles and how it actually targets B cells, and so it affects the memory population, and so antibody production against other viruses. So sometimes there's an indirect virulence factor that happens with some bacteria and with some viruses. So again, with adhesion, viruses are going to have molecules that act as ligands to bind receptors on host cells, just like we see in bacteria. And so some of these adhesion molecules, this also lends into anagenic variations, come in different forms. So you can see with herpes simplex viruses that we've got these glycoproteins, but we have different forms of that glycoprotein, and it allows for attachment to mucosal surfaces in the mouth or in the genitals, and so that we can get infection. But depending upon what expression there is, that means that if you've been exposed before and you do have an antibody response, it may not be sufficient against the glycoproteins, which this new variation exhibits. And so you could still get attachment. With HIV, there are glycoproteins. The one that's been studied most is GP120. And so that's going to bind to the CD4 and CCR5, which is a chemokine receptor, or it can also bind to CXCR4. These are receptors that are found on immune cells. So HIV is going to target specifically immune cells. And so when HIV targets the immune cells and destroys those immune cells, it's going to affect how the immune system can respond. It's not going to be able to respond as well to HIV, but also other pathogens too, because traditionally with HIV, the CD4 population ends up being affected. So you have a diminished CD4 population, and in some cases, when you've progressed to AIDS, you have no CD4 T helper cells present, and so they're not able to help in that immune response. And so by the virus targeting those cells, it has this indirect effect of affecting the immune response, and not only against that pathogen, HIV, that virus, but other viruses. And so with HIV, for some of these, it's a secondary infection that actually causes the complications, but it's due to that primary pathogen, the primary infection. With influenza, influenza has hemagglutinin, so hemagglutinin is able to bind on respiratory and intestinal cells, and so it's able to then affect those, and then you get signs and symptoms such as respiratory illness or GI illnesses with influenza. The hemagglutinin, there's a number of different variations of hemagglutinin. There's also neuraminidase, and so when we look at influenza, it's usually characterized by H, some number, N, some number. And so H1N1 is the first hemagglutinin, first neuraminidase characterized. And so that's actually back to the 1918 flu that was pretty widespread. And so again, from there, additional hemaggglutins and neuraminidase have been identified. These hemaggglutins bind to specific receptor respiratory cells and intestinal cells in specific hosts. So like H7N7, that tends to infect horses and dogs, doesn't tend to infect humans. And so the hemagglutinin and the neuraminidase do not bind to receptors that we find on human cells. More commonly, again, H1N1. There's also H3N2. Those are the common human strains, and there's a couple other strains. So when we look at anagenic variation, what can happen is we can get what's known as anagenic drift or anagenic shift, okay? So with anagenic drift, this is where there's small changes to those hemaggglutins and neuraminidase, and those small differences happen over time just because of random mutations, as just like in our own cells, as we replicate and use the genome to make proteins. There can be small error to make the proteins, but to replicate the genetic information, there can be small errors due to the polymerases that are being used. And so again, we can get this drift, this changing of the neuraminidase and the hemagglutinin. Usually these changes aren't so profound that it changes like the neuraminidase and hemagglutinin numbers. So it could be just a subset, a substrain that ends up arising from a drift. A shift is where we have a major change in the hemagglutinin or neuraminidase, and so this makes a new strain, a new novel virus, if you will. And so you can see here how we have these two different viruses. So virus A and virus B, they have hemagglutinin and neuraminidase, but these are unique neuraminidase and hemagglutinin for each of these viruses. But then we end up with virus C that has hemagglutinin from virus B and neuraminidase from virus A. So again, this is a new virus, so it's a major shift. It changes it. So usually with pandemics, influenza pandemics, it's because there's been some shift and we don't have immunity, we don't have protection against that shift. So the antibodies that we have because of what we've been previously exposed to, so if we were previously exposed to virus A, we would have antibodies against that. We would have T cells and B cells, memory cells against those. But when the shift happens so greatly that now we have exposure to virus C, those antibodies aren't going to be sufficient to fight that off. So again, generally with pandemics, we see shifts happening, and that's what we've seen with the coronavirus, right? So it's not that coronavirus hasn't been around before, but there was a major shift in those surface proteins that bound to ACE2 receptors in that spike protein that caused it so that even if you'd been exposed to coronavirus previously, it didn't give you protection against COVID-19. Exposed to drifts, which are just subtle changes, so sometimes those antibodies that you had to the previous one would still work. Now, looking at some of the miscellaneous virulence factors, they're very similar to the other ones that we've already talked about. So for fungal pathogens, for virulence factors, they can produce capsules, so similar to the bacterial capsule where it affects phagocytosis. Also, fungus can produce toxins. These fungal toxins are known as mycotoxins, and so they can disrupt the tissue, so they can cause damage to the tissue, but they can also invade the immune response. So again, we looked at that protease in the Journal Club paper, so that would be an invasion of the immune response. Now, they did talk about in the paper how that protease is known to break down extracellular matrix, so it's possible it's also causing disruption to the healthy tissue, but that was not the focus on the paper. The focus was on how it cleaved chemokines and how it cleaved the colony stimulating factor so that you wouldn't get activation and you wouldn't get recruitment, but then you also wouldn't get activation of immune cells to location. With protozoans, they can have a number of different virulence factors. So they can have adhesion molecules. They can also produce toxins. They're also good at anagenic variations. Also, due to their life cycle, their anagenic variation, they naturally have anagenic variation because if they're in a cyst form versus adult form, they're going to have different antigens, different surface molecules. And so, again, depending upon what the individual's been exposed to, what stage of the life cycle, it can affect whether you have a prior immune response to that. And they also have the ability, especially in the cyst form, to be able to survive inside phagocytic vesicles. So even if they've been engulfed, they can survive, and they can even germinate into adult stage. For hemolyphs, again, they have a number, just like protozoans, they have a number of different factors. One of them is just they're larger, so it's going to be more difficult for the immune system actually to take them down. They also have virulence factors that allow them to get entry into the host cell. So some hemolyphs are going to produce digestive enzymes that are going to break down tissue, so like the skin, so they actually can get in through the skin into the host tissue. And now they're into tissue where it's a little bit harder for the immune system to get to. They will also hide in places that have immune privileges. And so there are examples where there are hemolyphs that can get into the brain, and so there's individuals that have tapeworms in their brains, and they'll actually form almost like an abscess around them, and so they're protected from the immune response. Not a great life for them because they just stay kind of in this dormant stage for, and again, it works because they're not causing damage and the host doesn't attack them. There's a number of ways that they can evade the immune system besides some of the immune responses that they elicit, but they also have structures like the cuticle. So a lot of hemolyphs, they're going to have a outer kind of shell on them, and so this tough outer cuticle is hard for the immune cells to attach to and engulf. Hemolyphs are also able to change their polysaccharides that are on their surface. And sometimes their polysaccharides can be similar to the host. So this is known as glycogen gimmickry. So kind of a take on mimicry. So they mimic the glycogens that their host will produce, and they also can change them. So just like with anagenic variation, if you change those polysaccharides, responses that had been to those polysaccharides previously would not work. They can also change protein expression on their surfaces. Also, they can produce proteases that are going to degrade the host antibody. So even if you do have an antibody response, so keep in mind a lot of times hemolyphs, parasitic worms are going to be found in the gut. The gut's going to produce mucus. In the mucus, there are going to be IgA antibodies, and so again, they produce proteases and deactivate those antibodies. So they're not affected. They can have an effect on cells that produce mucus, so the goblet cells also. So it actually gives them protection against the immune response. And again, they can produce other substances that are going to suppress both innate and adaptive immune defense. So this concludes the audio lecture for introducing you to microbial pathogenicity and some of the virulence factors that different microbes can have. So definitely, if you have any questions, please feel free to ask. The OpenStax textbook is a wonderful source. The CDC also has a number of sources on different pathogens, both foodborne illnesses, so bacterial, viral, but also some of these other, fungal protozoans and hemolyphs, and some of the different research on it. Also, you can always do a PubMed search and put in your favorite pathogen and virulence factor and see what are some of the virulence factors that your favorite pathogen has. Because again, this is just to give you an idea of some of the different virulence factors. It by no means touches on any of them. One of the aspects I didn't talk about is kind of the genes themselves and where do we find genes. So virulence factors tend to be coded on plasmids. So those are that small circular piece of genetic information in bacteria that's kept separate from essential genes. We also see them found in pathogenicity islands. So these are areas that have high GC ratio. So if we look at a genome, we can look at the GC ratio to indicate, usually with virulence factors, bookended on either side of the gene are going to be these high GC ratios. Pathogenicity islands are more likely to be able to be moved from one location on the chromosome to another location on a chromosome because of those high GC ratios. So again, I didn't really touch upon that aspect, and maybe that's something we need another audio lecture for. So if you want an audio lecture on the genetic aspects of virulence factors, just let me know and I'm happy to put that together.